Hello, and welcome to another video. And today we're going to be looking at the cell cycle. So, there's three primary reasons why cells reproduce, which is the main purpose of the cell cycle. So, first is binary fission, and it is when prokaryotic cells, and if you remember those are the ones with no nucleolus, to split a bacterial cell into two identical cells. That's basically how they reproduce asexually. Humans use it for growth, repair, or development. So basically, if you are a human and you get like injured on your arm, you don't have a hole there forever. Like your skin will eventually heal itself and recover. However, there are some cells that do not do this. For example, if you got whacked by a hammer on the spine, you would suffer permanent damage and likely paralysis there. This is because neurons are usually in the G0 phase, which means they don't experience most of the cell cycle and thus like don't reproduce. And so that way, the injuries on your spine would never be recovered because they don't undergo cell reproduction. Next is sex cells, which are basically used to reproduce sexually. So every time a cell reproduces, it follows three steps. First, interphase. So 90% of a cell's life is spent in this phase. So interphase itself is divided into three phases, so we'll look at them individually. So first is G1, which you can see on this diagram, is the first phase to occur after mitosis over here. So on this part of the cell cycle, the cell will rapidly grow and grow bigger because remember, it's a new cell just formed from mitosis. So it will rapidly grow bigger and like prepare to become an adult of its own. It's like how children grow way faster than adults do. Next, normal cell functions are carried out. The cell like performs its like duties. If it's like a heart cell, then it will do the functions necessary, and it begins to make preparations for cellular division. But however, in this part, it's mostly focusing on increasing its size to prepare for cellular division. Now, in the S phase, which is over here, it's where the DNA inside the cell, which is an interphase, starts to replicate and get ready for mitosis. Because if you remember, in mitosis, one cell splits into two cells, so it needs to replicate everything. So and basically, the DNA will replicate so that there's enough for both cells. Next is G2. So in G2, it gets like the final steps done, ready for mitosis, and like it checks if everything is all good, if DNA replicated properly, and then after that, it will undergo mitosis. Another thing that occurs in G2 is all the main like organelles inside of the cell will turn into like a gooey like cytoplasm. Basically, like they'll turn into like the liquid around the cell, and then the liquid will separate into the two cells during mitosis, and then will reform back into organelles during the G1 phase. All right, synthesis, DNA replication. It's the process of making a copy of a piece of DNA. It occurs every time a cell divides to make a new cell, and it is also divided into three steps. So we'll look at each of the three steps individually. So the helicase unwinds and separates the two original strands to create a replication like fork, I guess you could look at it. The DNA joins into the strand and adds complementary nucleotides to each strand. So if you remember, nucleotides are the macro they are one of the building blocks of nucleic acids, which are a macromolecule. And basically, if you look at the double helix shape, these things, like over here, those would be basically what they're talking about whenever they're referring to pairs. Next, ligase glues the new nucleotides together. So basically, like I said in the double helix model, these two things would be glued together, forming two DNA molecules that are identical to the original. So basically, that would happen twice because it's replicating. And circle the enzymes below. I'm sure you can do that on your own. So, since a eukaryotic cell can add 50 nucleotides a second, prokaryotic cell can add 500 nucleotides per second. Do errors occur in this process? Of course. However, DNA polymerase proofreads and eliminates most of the mistakes, so it like checks it and sees if everything is actually like proper and making sure everything is good. So it actually only has one mistake per 1 billion nucleotides. And as you can see, for us, they only produce 50 nucleotides a second. So you can imagine how few mistakes there are. And there's a bunch of other checkpoints checking for mistakes too. So you generally don't have to worry about mistakes occurring in this process. However, we'll get into some of those mistakes later. So, in the G2 phase, the nucleus prepares to divide. The organelles undergo the process I referred earlier, and microtubules form, which are basically parts that like make up spindle fibers. All right, cell division. 10% of a cell's life is spent in this phase, and it's generally mitosis for somatic cells and meiosis for non-somatic cells. However, we'll look at mitosis for now. So mitosis is set also split into a bunch of parts. So the first step is prophase, and it is when the chromosomes condense into the center and become visible. So like you would see like a bunch of like stuff like in the center of the cell, like grouped together like this, and like visible, like they would be standing out, and you would maybe see some organelles. Next is metaphase. So in metaphase, what would occur is all the chromosomes would line up in the middle of the cell. So it'd be like this, and then you could see like the spindle fibers attached to them. Next, as you can see by this, is anaphase. They move towards opposite ends of the cell. So like there would be some chromosomes here and some chromosomes here. And finally, there is telophase. So in this one, it'll use the moved away chromosomes to form like nuclear membranes. And so here there'd basically be two nuclear membranes. However, you have to remember in telophase, you can still see some of the spindle fibers connecting the two. Whenever the two are like, here, I'll just draw a plant cell as an example. So one of the two are like completely separate like this, and they're not in interphase, that's actually called a cytokinesis. It occurs right after telophase, and the main way you can differentiate the two is to see if you can see spindle fibers between the two or not. Alright, so this is mitosis. And the way you can remember this is IPMAT-C. So there's I for interphase, PMAT, and then C for cytokinesis. Alright, so this is a brief description of each of the parts occurring in cell division, and you can read over this if you want, but I explained it earlier, so I'll skip over it for now. So cytokinesis is division of the cytoplasm. So it occurs very differently in plant and animal cells. So in animal cells, there's like a cleavage furrow, so like the two cells will like look like that, and eventually they'll split apart, like as expected. And in a plant cell, there's a cell plate instead, which kind of looks like it's like splitting the two like this. All right. And that is it for this video on cell division and mitosis and interphase. Hopefully you ace your test, retest, whatever. You're going to ace it. Good luck.